I checked out my plasmid sequencing data from like a week ago now, and it was pretty low quality. Let's see what we've got on this morning. Good morning, everybody. My name is Sophie, and I am a first-year PhD student in genetics. This week I'm going to bring you inside the lab, show you what I'm doing, and talk you through why I'm doing it. I'm going to be doing some CRISPR, and I'm also going to be looking at some fluorescence. I'm also currently trying to learn Adobe Illustrator and InDesign. If you have any tips, I'd love to hear them. Let's see what we've got on this morning. So first things up, we're putting our media in the water bath. This can be really frustrating, especially when you're going in early in the morning. You go in half an hour before you even want to start something just to get everything warmed up. But I've talked to you guys through all of this before, so I actually want to talk about my 10-week report right now. So in my 10-week report, I have a really detailed look at what I'm going to do in this first year, and then kind of broader aims for what I'm going to do over the 3-year lifespan of my project. I did include a tiny bit of data, some of this fax work, and my molecular cloning strategy, but it's much more focused on the size and scope of my project. If my 10-week report said one of my goals was to cure cancer, this would be an opportunity for my committee to turn around and tell me, probably not. If my 10-week report said the entire goal of my thesis was to complete a successful PCR, my committee would tell me to aim higher. It's also the first piece of written work that I've submitted to my supervisor, so I really hope I haven't said something silly or written something out the wrong way. She is obviously an expert in this area and I am not, so it is especially nerve-wracking to be submitting my draft, talking about the scope of my project, the techniques and tools I'm going to be using, and the disease we work on. Trying to write about that, knowing an expert is going to read it, is a special sort of stress. So while I can't show you my 10-week report because it includes some sensitive data, I did want to explain to you guys what it is and why I'm spending so much time on it. Good evening, everybody. I got a few boxes ticked off today. I finally got my 10-week report sent to my main supervisor. I got my figure to a standard where I was comfortable to send it to my supervisor. I don't think it's done. I'm still working out how to make things look really neat and tidy and professional. And I got a bunch of tissue culture work done. I've frozen down some aliquots of my cells so that I can come back to them later. I've split some of my cells that I'm going to be transfecting on Wednesday. And then also just some general maintenance on the transfected cells, just keeping them refreshed and growing, ready for analysis on Thursday. I've just realized I completely forgot to look at the plasmid sequences, which I was definitely meant to do today, and then streak out the relevant glycerol stock this evening. So yeah, I completely forgot to do that, but I got a lot else done. That one can go on the list for tomorrow. I'm most relieved about having sent off my report though. There's a nervousness in that I want my supervisor to be really happy with it, but I also know that it's not perfect and that I want her input and advice on it. And I wouldn't be sending it to her to draft if I thought it was perfect. So yeah, I'm really happy with having finally sent that off. I think I put it off a bit longer than I should have, but I'll see you guys tomorrow. Good morning, everybody. I'm on my way in a bit later than I normally would be. I checked out my plasmid sequencing data from like a week ago now. It really got put on the back burner and it was pretty low quality. It looks like another bad extraction. So that means doing another overnight culture today. And generally you don't want to put an overnight culture on in the morning. You want to do it as late in the afternoon as you can so that tomorrow morning when I come in, they'll be in the right phase of growth for me to do the extraction. So I shuffled my bookings around a little bit and here I am. See you in there. Good morning, everybody. Welcome to Wednesday. When I'm doing transfections, Wednesdays and Thursdays are kind of the big part of my week. Wednesday because I'm setting up the gene editing, and Thursday because I look at the facts, so basically the results from the experiment I set up last Wednesday. So today is setup, tomorrow is results, but from last week, not today. I've also got my overnight cultures that I set up yesterday after the failed plasmid sequencing. So I need to mini prep those, and we're going to try a different way of screening them instead of sequencing this time. So it's a pretty full day. I better get started. Good afternoon, everybody. Today was a long slug and I did not get the results I was looking for. So I did a transfection this morning as planned and it was pretty good. I did have some arcing, but I think it was just one sample, so that's very acceptable. And then I did some regular tissue culture maintenance, just changing some media, splitting my cells for tomorrow, just routine stuff, and it all went well. But then we went to my cultures, so I mini prepped them. So I got the plasmid out of my bacteria. Concentrations were good, 260, 280s were good. So then I tried digesting them with some enzymes I have and running that product on a gel. So if my insert is in my plasmid, the enzyme will be able to find it and cut, and that will change how these products look when I run them on a gel. This process took hours, maybe like 40 minutes for the DNA extraction, 30 minutes to an hour for the digestion overall, and time making a gel, more than an hour running the gel. One of the gels didn't set correctly, so I had to throw that in the bin and make another gel. So then I had 2 gels running. One of my gels then melted in one of the tanks, but I still imaged it and kind of got something out of it. So after all this work to then find out that it doesn't seem like there is an insert in my plasmid and the banding that I was expecting didn't show up, that's pretty frustrating. I've decided I can't do much more with it today. I'm gonna head home. Tomorrow I'll culture them out again probably and try to repeat this to some extent, but I just wanna step back and think, is there anything I'm missing before I jump into this and start rushing over just repeating it. Very possibly I've made a mistake, but it's also possible something's going wrong in the plan, and I need to assess that possibility before I start wasting reagents just doing it over and over again. So it's time to head home and think about my experimental plan and where to go from here. See you guys tomorrow. Good morning, everybody. Yesterday was both disappointing and confusing, but we're gonna get it sorted out today, or actually tomorrow. But we've got to do some setup today to get to that point. Also, I still have my facts analysis on from last Wednesday, so we'll be doing that today. I'm really excited to see this analysis because I think only one sample arced last Wednesday. I'm gonna have to check my lab book on that, but I'm really excited to see like the most representative results yet, I guess. So we've got a lot in the diary today. Time to get started. Good afternoon, everybody. Good afternoon, everybody. I am heading in for my Friday. I had actually planned to do quite a lot of experimental work today, not just the usual set up the cells for the weekend so that they don't overgrow. But for the first time, I actually haven't really been able to get a lab booking just because of our COVID system. Not bad for working here for like 8 weeks now. This is the first time it's actually stopped me going in. And theoretically, I could book to go in at like 4:30, 5 o'clock tonight if I wanted. But even if I did choose to do that, I would then be working for an hour or two to do what I need to do. So then we're in at like 6 or 7 p.m. And I'm gonna do plenty of late nights and weekends during my PhD. I don't think I'm that rushed for these results right now. So yeah, because of the booking system, I'm just gonna be heading in for a very quick session to put my overnight cultures from last night into the fridge so I can check them out on Monday. Good afternoon, everybody. It is Friday afternoon. I am on my way home. I have a draft back from my supervisor, so I've got plenty to be getting on with. I think she said she wants me to add some more figures, a few more references, things like that. So we're on a good track, but there's more to do there. I'm also going to start planning my week next week on what I need to book and what I'm going to do. And I'm going to try and make a video about that. It is definitely hard to make a video about my lab book because my lab book has a lot of information in it that I shouldn't really be sharing with the whole internet, but I'm going to try. I've got a few plans in place for next week, which I'm already really excited for. And my 10-week report draft is due next Friday, so there's plenty to be getting on with. If you've enjoyed this content, please subscribe to see more of it. Leave a like and comment if there's something you'd like to see more of. See you guys next week.